Okay, I think we're back.
Yo,
Uh,
we're back.
yeah, I think we're back. Now, this is comedic and sad at the same time. I don't know. Like, I literally streamed yesterday for 4 hours continuously. No problem. 4.5 hours continuously. Zero issues whatsoever.
Huh.
I've adjusted my bitrate in the hopes that maybe that solves it.
Okay. Cool. We'll go for it.
Uh,
What do you think, man? Sorry, I don't know where
Yeah.
would you get?
Yeah, so where I got cut off again, unfortunately, is like you were talking about nuclear, and like you were talking
Yeah.
about how it seems to be like there's two sides of the spectrum, where it's like
Mhm.
on one spectrum you have people like Scott who is like pro nuclear, where you
Yeah.
can't really consider yourself part of the green energy or the renewable energy movement unless you're like pro nuclear. And
Yeah.
then there's the other camp, where it's like it's just like no nuclear no matter what. And
Mhm.
I think the last thing you were saying is like unless there's some sort of like major breakthrough, you can't really. It's hard to consider nuclear as an actual renewable energy source.
Yeah, it's not, right? It's just not a renewable. The source of the energy is a finite amount of whatever the element is. So by definition, it's a finite amount, and so
Yeah,
it's not renewable.
I agree with you.
Yeah.
And this is one of the things actually that maybe I have learned from the streaming space or being in this space.
Mhm.
Is like, so I have no problem admitting that I'm very far from an expert on nuclear energy. And mostly what I've learned is just through what I've retained from some of my undergraduate education in like physics many years ago. So outdated information, and also a little bit of just what I tried to keep up with in terms of the headlines. And it seems like so far from what I can tell, like my position should move. As in, like at one point, based off of what I was taught, like whatever 10 years ago in physics, like nuclear energy not really the best long term. As opposed to like now, based off at least what I'm hearing from the people who say that they're well informed in nuclear energy, that like, oh no, like really got to be like pro nuclear, and like the whole nuclear waste issue is like not an issue. That many of the things have been solved. As based off of what I got from your conversation with Scott, is that you lean more towards against nuclear energy. Is that like a fair assessment?
No, I mean there's times and places where it makes a lot of sense. So like for example, in Japan where they don't have the space for a solar or wind arrays. Sort of densely packed urban environment actually is really good for nuclear. Same thing with like large cities or something. So like the Northeast or something in the United States, like they don't have like all the space for all that stuff. So you can kind of like use nuclear to help out. But I don't think that it should be like the only thing that we're trying to do in terms of like renewable energy production, because it's not renewable by fact. And the problem is it kind of gets into this problem where it's like nuclear is not there yet, but it could be in the future. So we kind of like keep pushing off doing anything and investing into renewable energies, because it's like it's not there yet. And that's kind of like one of the paradigms I wish that people would shatter, and because my fundamental position is actually there is the technology right now to be 100% renewable in the United States. All of our energy, all of our fuel could all be renewable. We have it through algae biofuels. It's just using a smorgasbord of approaches. It's not just one thing, just wind energy isn't going to do it. But hydro with wind, with algae biofuels for the fuel. And so we have the technology now. So like, let's not just take this position where it's like, oh, we just need to wait till it gets developed. You know what I mean?
Right. Until it's actually like perfect or perfected.
It never will be.
Yeah,
It never will be, right?
Yeah, exactly. Don't let perfection be the enemy of progress, right? Is the expression.
Exactly. Yeah.
So now that we've been, I think, talking continuously for 5 minutes at least, do you want to give yourself like a
Hmm.
formal introduction, and a little bit of like your background, and you want to like share a little bit of your research?
Sure. Yeah, I'm Greg. I have a YouTube channel called The Peer Review. You can get there at thepeer.review. And I basically try to talk about the coolest things that are happening in science that nobody else is kind of talking about. And I think that there's a lot of cool breakthroughs and stuff that just don't get mentioned. I don't know why, because it's easier to be doomer about climate change. It's easier to talk about how bad things are, I feel like, as opposed to like some cool breakthroughs and cool things that are happening. So I mean, I started in an algae biofuels lab in 2009, in where I worked on a paper making cancer-killing algae, where we killed non-Hodgkin's lymphoma stage 1 in mice with immunotoxins made in algae chloroplasts. And then I
Okay.
worked at a plant biotechnology company for 5 years, doing plant um, gene editing, and then I did yeast gene editing, and then I worked with next generation sequencing, and then now, like right now, my current research is, sorry, see if you hear my
No,
baby
no,
girl
that's
in the
okay.
background. Um, um, right now, I work with nanomaterials and nanotechnology, trying to facilitate and kind of bring that into the algae space for the first time.
Okay. If I may ask, so you mentioned earlier some of your work is like, if I remember correctly, generating immunotoxins to treat cancer in chloroplasts. That so, out of
Yeah.
curiosity, and so for me, technically, like my formal training is in biology, as in I did my bachelor's in cell and molecular biology. But like now I've
Okay.
moved more in the biochemistry direction. But out of curiosity, is there a particular reason why chloroplasts were like the main sort of bioreactor for these immunotoxins?
Yeah, because the chloroplast is prokaryotic, so it's a evolutionarily
Right.
through phagocytosis, it's been taken up. So the cyanobacteria was taken up, so it actually has prokaryotic genetics. So and large proteins that are created in the chloroplast get encapsulated in there. They can't leave the chloroplast because of the double membrane. So it actually gives us unique ability to make things in a chloroplast that are one large, because there's a lot of like scaffolding proteins that are really good in the chloroplast, because it's used to making these large arrays of chlorophyll. And then two, it allows us to make things that would normally be toxic to eukaryotic organisms.
Okay, that's
But
what I was gonna ask. Like, why didn't you just use the cell line to generate whatever protein
mhm.
substance? Is that like pretty much
Yeah,
the main
so
reason why? Is because like yeah, you just kill the cells in the process?
exactly. So
Okay.
yeah, the immunotoxin basically had a little link to it, something that would kill eukaryotic cells. And if we made it in the chloroplasts and then extracted it, then we could deliver it to eukaryotic cells that would eventually kill it.
Okay, yeah, that makes total sense. Because yeah, as soon as you like describe that, I was like, well, of all the things to choose, why chloroplast? There must be a reason
Hmm.
that I'm not seeing, and that makes like perfect sense to me. Um,
Hmm.
and so like it seems like at least based off of what you outlined, you do a lot. From what I would understand, you do a lot of like gene editing, and like pretty much like genetic engineering of organisms to produce like what you want.
Yeah, yeah. I'm like, I guess my training is a lot in like a molecular biology. My
Is it
undergrad was in like ecology, but I've done like practically for like 7 years molecular biology. So changing genes, and at parties, I'm like, I genetically modify
yeah?
organisms
Are you
for
like
a job.
essentially more in like the synthetic biology realm? That'd be like the more, I guess, technical term.
Um, I guess like it'd be like genome engineering. I see synthetic biology. I'm doing more of that now, kind of like creating tools for synthetic biology, like delivering gene cassettes, delivering like protein expression and all that kind of stuff now. But like before I was working with like genomes, so like changing the genomes, creating trying to create product A. In order to make that product, then we need to like knock out a bunch of pathways so that all the resources are shuttled into that one pathway. That's the larger goal.
Okay. And so it seems like, again, going back to your conversation with Scott, is like right now your work is in biofuels, and you're pretty much like all about the algae biofuels.
Yeah, I mean it's
Welcome to
kind of
Flavor
like
Country.
figuring out ways that so like
Oh,
nanotechnology and
sorry.
microbiology.
I have to tell you. So someone came into my chat and told me to tell you that your audio is busted on your stream.
Oh, okay, interesting.
Whether you want to address that or not, I don't know, but I feel like I should let you know that. You've been patient with my
Okay.
technical issues, so I will
Oh,
absolutely
thanks.
return the favor in kind.
I wonder why. Let's see. I was trying something different with my audio. That's I'm like, oh man.
That's a lesson from running experiments. As soon as you get it working, do not change anything whatsoever.
I know, right? Okay, let's try this. Okay, I think that these are my old settings. Like, so maybe this will work. We'll see.
I see no reason why not. If old settings worked, they should work.
Yeah. Yeah, so like I'm kind of, so the world of microbiology is basically like 99.9% undiscovered. Like we can't really work with 99.9% of the organism microbes because
Yeah, the Great Plate anomaly.
Yeah, we can't grow them. We can't genome engineer them. We can't change their genes. So the idea is to try to use nanomaterials, nanotechnology in order to deliver genes and deliver things into the organisms as they are. And that's the idea. So using so now with these nanotechnology as like a carrier, like a tool, then now we're able to kind of like synthetic biology is basically removing that biological black box, creating ways to express proteins, see if proteins and genomes and are present without kind of like but the problem is that basically the community of microbes that kind of exist and correspond and work with one another creates like an inability for us to. It doesn't work like to just culture one organism. It doesn't work. That's why we can't culture them, is because they kind of need to be in that community. They need to be signaling. So synthetic biology can kind of go into these microbial communities for the first time, kind of see what's going on as they are. So that's like in situ research. So where they are as they are. And that's why nanotechnology is kind of like an interesting platform in order to explore synthetic biology techniques and research.
Yeah, and this harkens back to, again, my undergraduate days, where like, yeah, I've taken a couple of microbiology courses, and that's like the one thing, that's one of the main messages that I remember is like, yeah, in terms of the existing number of microbes out there or bacteria out there, like the percent that we actually like know of or are able to like culture in the lab, it's like just like this tiny sliver, where like most people think, understandably, they think that way, but like most people think like, yeah, if you want to just culture bacteria, you just like take a streak and like streak it on a plate and then like it'll grow, but it like virtually never works that way, and that they very much need their specific environmental conditions, and from what I understand, very often, like even in a lab where we can actually like simulate, or we get close to simulating their like native environment, like they just will not grow for some inexplicable reason. Where it's like, yeah, on paper, like this should match their native environment, yet they're still not growing, and like we don't know why. But like,
yeah, it's a there's nutrients involved, right? They have to have like specific micronutrients, but then they also are chemically signaling to one another to make sure that they're still present and they're still existing. So when you kind of remove a single microbe from that environment through plating or any type of selective pressure, you create the community collapse of all those microbes. So and it's so important to figure out microbial communities, because we're finding now with microbiology research that microbe communities are everywhere. So they're in your gut. That gut is connected to your brain activity. It's connected to your immune system and T and B cell production. You have root microbiomes, and plants are incredibly important for nutrients and productivity. So plant productivity is tied to root microbiomes. The plants and trees actually communicate to one another and communicate like what's happening through these microbial communities. So they actually talk to one another. Plants talk to one another through this stuff. So it's really cool.
Yeah, no, like the microbiology courses that I took, like I enjoyed them immensely. And like I have no doubt that my information is like outdated anyway. So I have no doubt it's only gotten cooler and cooler over time. And so out of curiosity, and actually, if I ask anything that you want to answer, feel free to like say you don't want to answer.
Go for it.
But it seems like you spent a decent amount of time, or a fair amount of time, basically from your undergraduate to I think you're pursuing your PhD now, where you spent some time in industry. Was it always your plan to pursue a PhD, or as a result of your experience and like your passion has grown over time that like, oh, you felt now you really want to pursue the
when I left undergrad, I loved the academic environment. I loved that the idea that you're working with a team and you're working towards something that's bigger than yourself, and you're trying to solve some of the biggest problems in the world that exist. And it's just like, wow, it's so cool. I didn't have no idea people existed like this, or there's groups of people that exist like this. And it really made me want to be a research scientist, but I kind of wanted to make sure that I loved science before dedicating my life to it, like actually doing the science. So by going to the biotech, it kind of allowed me to see kind of like the everyday of like what science is about, and do it with a 9 to 5 as opposed to like the life commitment of a PhD. So I was still kind of exploring that idea. And then, once I got to biotech, I was like, wow, this is really something that is fun. Like this is, the actual science. I love doing it. And then CRISPR came out too during that time. So I'm actually genome engineering Erolia lipolytica to do this certain thing. And then all of a sudden this like new gene editing tool, it gets released on this Saccharomyces cerevisiae paper. That was like the big paper where people were like, whoa, this actually works outside of bacteria. This is kind of cool. So I was like, oh, I'm going to try this. I'm going to try this new CRISPR-Cas9 thing in my yeast. And, that was, nobody had done that before. Nobody had used CRISPR in Erolia lipolytica for that time. And then, so that was like a year-long research thing at where I'm trying to do that for the first time, and kind of seeing like, dang, this cutting edge research stuff is really, really addicting. So it was that point where I was like, you know what, I need to go back to get my PhD, and I want to dedicate my life to science, kind of thing.
Okay. And do you feel like your time in industry like has given you like a step up, or like basically do you feel like it's been a smooth transition into your PhD as a result of like your industry experience, or like now that you're in the PhD and pursuing it, like, oh, now you see the value, as in like, oh, there was some like, do you feel like there were some like glaring holes in like your knowledge or skills, technique, that has come to your attention throughout the PhD, or like basically like it's a lot of the same, it's just now like in an academic setting?
It's interesting. Like there's so much consternation that professors had about me like working in industry. Like I had professors berate
that's
me.
the devil, right?
And try to exactly
evil corporation.
It like to a lot of professors, you got to realize that like science is like the Force in Star Wars, and you're literally like you're Darth Vader to them. So you're like, dude, I'm on the Death Star. I'm an engineer on the Death Star, but I'm doing really good engineering, and to them, they're in the Jedis. You're the Jedis, and they're like, I don't give a damn how good of an engineer you are. You're still working on the dark side, and you're working for the Death Star, man. That's like, to them, there's nothing that could that's good about it.
You're still the Umbrella Corp from like Resident Evil, and you're
Exactly.
waiting to release the virus, the plague
so
upon us.
I had professors get up to me and be like, how dare you think you're like in a biotech, a plant biotechnology? What do you think about Monsanto, huh? What do you think about what's happening out in India? And I'm like, dude, this is a graduate school intro panel. Like this is so great. I told one of the graduate programs, I was like, that guy was so unprofessional. He blew his lid like in the middle of my interview process, and I was like, this is crazy. So I think that like, and then also like this, a lot of professors, like how do you even think, are you even capable of doing hypothesis driven research if only you've done,
For
stuff
profit
in
research.
yeah, exactly. Do you even know what science is, kind of thing? And I've really thought about that. Like, can you become like a scientist, just by working in industry? Can you do that? Do you need to get a PhD in order to like become a scientist? And I really fall down now, like on the side of like anyone can be a scientist. About it's just how you present your ideas, how you like research them, how you test them. And especially now having kids, like they have observations all the time and they make hypotheses all the time, and we test them together, and it's just really I don't know, it's just really rewarding and stuff. I don't know, and so I do think that you can do really good science in industry. I think that industry it's really interesting. Industry really focuses on reproducibility. So and in academia they really focus on publishing. So like for one, so it's like and that's the peer review process. So do you which side do you think is more important in the scientific rigor, right? Reproducibility or the peer review process? So I think that it's kind of interesting. I mean, because in industry, you're not going to do something just once. You're not going to do it twice. You're going to do it 20,000 times a week. So you got to make sure everything is reproducible. You got to write down every standard operating procedure. You have
the SOPs.
Oh yeah, exactly. If you're not writing it down and you don't have a live notebook, then it's just absolutely dead, right? On the other side, you have, sometimes, I mean, in grad school, you got people that are going into a procedure or a method that they have know nothing about. They've read it a couple times. They put the right controls in based off of other stuff, and they'll do it once, maybe twice, and they'll put it in a publication, and Yahtzee, done.
Yeah.
So it's like, whoa,
it's worked once. Let's just go with that.
Yeah, exactly. The controls worked, everything's worked, and you're like, whoa, dude. So, like, do you value reproducibility more, or do you value the peer review process? I think that there's, but yeah, then again, the peer review process with academia is rigorous. You have months and months of people, ripping your experiments to shreds. Your manuscripts are, hundreds of comments every time that you submit it to your authors, and people are saying like, this doesn't show what you think it says. This sentence, it needs to be ripped out. You need to completely rewrite this whole section because you're not proving anything here. So it's interesting. There's like a lot of each side is very different. I think I've going through biotech, though, just has given me more of a familiarity with the tools that are out there and reading research and all that kind of stuff. So I think that was good. But yeah, different ideas, different emphasis on each side for sure.
Yeah, absolutely. And it's actually interesting because I'm almost sort of, when it comes to experience, the mirror image of you, in terms of like, I pretty much like spent the vast majority of my time in academia, as in like from undergraduate to graduate, to my now present. But then as I've sort of gotten deeper into my PhD work, I got a job at a biotech startup. So it's only in the past like year, almost exactly year and a half, that I have some biotech or industry experience. And yeah, it's totally interesting, sort of contrasting the two in terms of like how science is done, one versus the other. Not to say that one is necessarily better, but it's sort of like, although I'm now sort of leaning more towards one as of right now, but like, so to be transparent, I I stayed away from industry for the longest time because like I didn't want to work for industry. As in like my original plan when pursuing graduate studies was to, get my PhD, do a postdoc, and then hopefully search for some sort of faculty position, maybe teach,
Yeah.
ideally both. But now that's no longer my aspiration. Where it's like, yeah, I no longer want to do a postdoc, and in fact, like I'm pretty committed to not staying in academia. So once I came to that realization, I was like, oh, I'm gonna start like maybe pursuing opportunities while I'm still like doing the degree. And sort of an opportunity just kind of like fell in my lap, which I'm grateful for. Or like a colleague graduated and they started working for a company and they like threw my name in the ring.
Nice.
I was like, sure, yeah. Well, I mean, it worked out, or I was, I guess, lucky in the sense that I had a certain amount of expertise that appears to be difficult to find. Or maybe they're just not looking hard enough. I don't know. But, it's super interesting comparing even just like the simple stuff. As in like, yeah, so, science at university. So I'm in Canada. I'm in Quebec, Canada to be specific. But I think it's hold true in the United States where like, yeah, academic research, it's like publicly funded. And so like me as a graduate student, the science that I do in the lab, that's funded pretty much almost exclusively by, taxpayer money. And I don't blame any taxpayer for not knowing how science is done, but like, I don't come from a well-funded lab, and so we have to do some cost saving. Whether you're familiar with that, I don't know.
Oh yeah.
Yeah.
Oh yeah.
And so it's like, as of today, I'm reasonably confident that I'm not gaining any more knowledge or new experience when I'm like making my buffer and like adjusting its pH. Like I think I have that down pat pretty good. And I've had that like on lock for a long time. Pretty much I would say since like day one, maybe day two. But like, it's pretty much like a waste of my time at this point to do it. But since the lab has to save money, it's like, well, yeah, we're gonna have you like make the buffers. We're gonna have you like make your acrylamide gels, as opposed to
Dude, that's dangerous. That's crazy.
As opposed to like with a biotech startup, it's like we're just gonna buy the buffer. Why we're paying you too much to have you waste time making buffers.
Yeah.
And like that's something that I appreciate. Not that it's like all sunshine and rainbows in the industry side, but it's sort of like I feel a little bit undervalued when it comes to like my academic research. Not that I would say that I feel perfectly valued in industry, but at least there's a more recognition of like, oh, okay, this person has expertise, and now let's actually like leverage their expertise. Let's not have them like waste time filling up tip boxes.
Yeah.
Not to denigrate anybody that does that work, but like if the taxpayer learns that, hey, you're funding my PhD, and like I'm filling like tip boxes and using the pH meter to adjust my pH of my buffers, it's like, well, what are you doing? This is like a waste of your time. Not only like it's making process longer than it has to be. Has that been somewhat a similar experience for you?
Yeah, man. I mean, I tell people it's like, academia, they want you to shoot for the moon, but then they give you like the used Amazon shipping boxes out back. So it's like
maybe a jetpack.
Yeah, you're duct taping stuff together, and it feels very startup-y kind of thing, or at a small lab that's like not super well funded, because like you're doing 1,000 different roles, you're kind of involved in everything. Professor's job in the United States, it's like basically an independent contractor where they're responsible for their own funding. So, and then on top of that, they're kind of like playing the CFO. So they have to like beg for money. CEO, they're the chief executive, making all the hiring, firing decisions. CTO, they're like reading papers, trying to grab the new technologies to bring into their lab. So they have a lot of different hats that they're constantly doing. But at a biotech, it's just like more individualized. Like I'm not, as a grad student, I'm begging for money. And at the biotech, the CEO's off begging for money. I don't need to go do that. So that's kind of like it's kind of nice. You know, I'm dealing more with bench work. I'm dealing more with the science. I'm in the lab more. As a grad student, you're everywhere. There's 10 times the amount of things that you're trying to juggle, especially, and then on top of that, teaching on top of that, which is kind of a its own thing on top of that, which is
teaching, grading,
a lot of time.
Yeah,
a lot of time. Yeah, I mean that's half of your week, and so it's very interesting. I mean, how like do they separate like research institutions and universities in Canada, or the art like all the research institutions basically universities as well?
So generally speaking, most of the research that is done, like we do have research institutions, government research institutions here in Canada, but they're nowhere to the scale of the United States. Like we have the NRC in Canada, that's the National Research Council of Canada. And there's a couple of others like similar institutions. But like generally speaking, if you want to do kind of like that fundamental research, of like, there's no direct application, or like there's only an application, far down the line. It largely takes place at universities. But there is a lot of good science. Like, I mean, that is kind of the dream for many scientists, at least here. Like, I wish that there was as much of a research institute infrastructure in Canada as there was in the United States.
I really feel like research institutions should do the research and that universities should just teach. I don't know. I really feel like that. A lot of professors will say like, oh, well, the whole point of college experience is to have the undergrads taking classes and being involved in the research. But it's like, I feel like, why can't you just like go to a research institution for once a week and do your research, and then come back to the university and the college and just focus on school and focus on learning through a classroom. Because the dirty secret in a lot of universities is that they're only there because they want to do research. They don't actually like teaching.
Absolutely.
If you talk to them and you talk to professors, you have a beer with them at the bar, they're going to be like, oh man, I got to teach this quarter. That sucks. Like, it's like, sorry. I mean, that's crazy. It's crazy that people are paying an undergraduate, especially with the tuition fees and all that kind of stuff, and they're not getting someone that's 100% tuned in. And I'm lucky enough to TA for someone this quarter that's like, you could tell that she loves teaching, and it really just completely changes the game. And you can tell. I mean, you can just tell. I just don't think that the undergrads should have to pay for
pay
a
the
tuition.
price. Well, yeah.
Yeah.
Absolutely. I agree wholeheartedly. As in, I think just about everyone knows that, like, if you have a professor that they don't want to be there, like, it. And, well, not just professor, just teacher. Any sort of educator. Yeah. If they don't want to be there, like, it's readily apparent. And yeah, that is totally one of the challenges that I have faced throughout my degree, of like trying to balance those things of like, yeah, as part of my degree, and also like part of one of the ways to support myself is through like teaching, being a teaching assistant. Where it's like, yeah, if I want to be able to like, have some semblance of ability to pay my bills, like I have to TA. And I love it. I actually do love that aspect. As in, like, I would love to be able to do it more. If there was like some sort of full-time position devoted to that, like I would do that. But the reason why I don't like it is because, like, yeah, it technically detracts from what is supposed to be like my main responsibility. Or like, I'm now like crunched, or like, I'm really have to cram in as much research as possible into a day, especially if I know it's a day where the type of TAing that I do, like supervising students in a lab, and that's like a 4-hour block. Then like some office hours, so like that's usually like a good 5 hours of the day devoted. Like yeah, my day is like shot, and it totally is just an extra added like stress and burden. But it's not due to the nature of the teaching. It's more like, yeah, I'm trying to like balance both things, and it's maybe I'm just bad at it, or maybe I'm just lazy. But it's like,
no, I don't think so.
It's it's a perpetual challenge, and it shouldn't be that way at all.
Yeah. Yeah, it's weird. I mean, there are benefits to, having, undergrads in the classroom where they can kind of like talk to you, and then they can see if they're interested in research, and then they can kind of like make those connections. And I mean, that's how I started, right? I started at an undergrad, level too. I was, cleaning glassware for their
Wow, okay, yeah.
experiments and making media and stuff for them, until I begged them to like teach me molecular biology and get into this project. So, yeah, it was. It's tough. I mean, there's just a lot of it isn't paid. A lot of it is free internships, where you're just kind of like going as an undergrad, and you're not you're not getting paid for your time, which makes it really tough. It's hard to, like, in classrooms and like how we teach science. It's just so frustrating,
It's bad.
because
Yeah, I've said
we're not
many
really
times.
teaching. We're not really teaching science. We're teaching, like, it's like bowling with the guardrails up, where it's like
Good analogy.
it's supposed to it goes, you hit the pins, but really, was there any other outcome that was going to be there? So we kind of like, we don't let them fail. And that's what's so frustrating, is that like failure is 95% of research science, where it's like it's the whole point is that it's never been done before. So the first time you do something, you're not going to get it done. I mean, you're going to have your controls work in a properly, planned experiment. You're going to have, the thing that you want to happen through the positive control, but now your experimental samples. So it takes, years in order to do that. For my CRISPR project in Euroia, where it hadn't been done before, it took me 1.5 years in order to get, CRISPR editing inside of, Euroia. So it was extremely difficult. It makes you think about it personally. Like, am I the problem? Is there something wrong with me? And
Absolutely.
you're looking through your sequencing, you're starting it from the square run. But that's why you go back and then you search through the literature, right? You search through the literature to create a hypothesis, test it, it fails, and then you search through the literature and go through that science. And we don't do that with science, and it's just really frustrating. Textbooks I feel like are really boring, just boring in general. I hate reading the microbiology textbook, and I'm a microbiology PhD candidate. It's like, I don't know, it's just very boring. I can't. There's no drama. There's, in a paper, there's like, for example, okay, I was just talking about this in this week in lecture. It's like, like the nucleus, right? Learning about the nucleus. Okay, textbook. All eukaryotic cells have a nucleus. It's like, okay, how about this though? Okay, that's a textbook thing. And the research side is bacteria, prokaryotics, organisms can bring in things from the outside world. And one of these things is, for example, the chloroplast is from cyanobacteria. The current hypothesis for where the nucleus came from is a large virus, because large viruses are good at encapsulating DNA. So the current hypothesis is that a prokaryotic cell, through phagocytosis, took in a large virus to be its nucleus. And it was tested recently in a recent publication where they actually amplified viral bits of genomic DNA inside the nuclear genome. So we actually have some evidence to show that the nucleus might have been from viral origins.
That's actually news. So yeah, I've been aware for a while of, the endosymbiosis hypothesis of, like, where mitochondria originates, where, as you said, the chloroplast originates. No idea about the nucleus. That is brand new.
But now you're gonna remember that story. You're gonna remember that. You know what I mean? As opposed to like, oh, every cell has. There's no drama. There's no like, what's the working hypothesis? What did they test? I don't know. I think it's better to learn fewer things and learn them deeper. That's my.
Yeah. I can totally get behind that. And I've said many times, or I've made it no secret that, like, even though I'm a scientist, and like, whether it's in my genes or not, I don't know. But like, I've considered myself like a scientist, or at least I've considered myself to have a passion for science from like a very early age, as in like elementary school. Of like, I literally had like a chemistry set growing up.
Oh, cool.
I had a little sort of starter microscope with pre-prepared
Wow.
slides, you know,
Oh, that's so cool.
to look at, you know, insect wings and all that stuff. Or it's like I started like tinkering with these things like before, you know, having any sort of exposure to science in like elementary school. And like I'm of the age where I grew up with like Bill Nye the Science Guy and the lesser
Oh
known
yeah,
and
me
the
too.
lesser known Beakman's World.
Oh yeah.
Very underrated. With the two
Hmm.
penguins, I think Herb and Dean and his rat. And like that's what I loved. And anytime that there was, I'm also of the age when book orders were a thing, where you know you got your little catalog, and you know I was always
Hmm.
picking the science books. But then when it came to my formal scientific education, largely starting in high school, like I hated it. I
Hmm.
hated high school chemistry, or back then it was called physical sciences. I did not like physics.
Hmm.
I didn't like, even though I went into biology, specifically molecular biology, I didn't like biology. I did not like, you know, looking at onion cells or like cheek cells, or like trying to identify whether it's, you know, anaphase or prophase or whatnot.
Hmm.
But maybe that's just me, where like it was just like super dry, and like I didn't really care for the, you know, experiments, my earliest chemistry experiments of like, you know, calorimetry and like just like whatever, boiling water and whatever, taking some temperature or whatever. Um, where it's like, yeah, this is like not what I imagined when I was like watching Bill Nye the Science Guy and Beakman's
Yeah.
World, or like any of the books that I happened to go through. Um, and like I'm glad that I like stuck with it to get to university when, in my mind, like the cool stuff happens. And
Oh yeah.
I understand why they teach it this way, because like to do the cool stuff, it generally requires like more money. Like to get to the cool stuff, you usually need cool instrumentation, and
Hmm.
uh, you're not gonna, well, most people would be against, you know, providing expensive cool instrumentation to, you know, high school students or even elementary students.
I mean, I agree and disagree. Like, I think that like you can teach the cool stuff in science without, you know, I do think that like there's some hands-on nature. You can have experiments. You need to have experiments fail. You need to remove those guardrails. But I think that you can talk to people about the cool stuff that's happening right now, as opposed to right now. It feels very like chronological, you know, where it's like we start off with like, you know, 300 years ago, 400 years. The same way we teach math, by the way. We teach math very like, you know, it's like very like through the ages kind of thing. Um, but I don't think that that's the way that it has to happen. I think that you can talk about the cool stuff that's happening and engage someone without having them to know like the lipid bilayer, blah 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 blah. The cytoplasm is a blah blah blah. You can just be like, man, all these organisms, all these living things, they have DNA, and we can change that DNA. And there's a code for life that will allow things to happen, and that's what differentiates everyone. Everyone has their own DNA thumbprint that's different. You know, it's like, um, you can like talk to them. Um, I think what you're going to point, and I totally agree, is that like the actual everyday work of a scientist is so much different than the classroom. Classroom genetics is totally different than like what you do at a genetics, you know, lab or at a biotech doing genetics. It's like, you know, like in 2010, like I took a genetics class at UC San Diego that was like we were still doing those stupid hierarchy charts, like
Oh yeah,
filled in
the
circle,
pedigree or
like
whatever.
yeah,
Yeah.
exactly. Dude, that was, and then the very last week was like, oh yeah, by the way, there's like next generation sequence happening that like I don't know, it's kind of like new. We don't really, it's happening. And then I go to a freaking the biotech, and they're like, okay, we're doing Illumina-based sequencing. We're going to align millions of different sequencing reads, and we're going to see if there's single nucleotide changes amongst 200,000 protoplasts in a cell. And I'm like, what? Like, I was just doing hierarchy, and I'm like, well, don't we? I made this stupid. I was like, what? Don't we have like, where's the hierarchy charts, right? Where's the hierarchy?
Yeah.
And they're like, dude, this company costs $3 million a month to run. Okay, you don't think that we're gonna spend $10,000 on a whole genome sequence for the organism that we care about? I'm like, dude, of course, duh. Like, why would we wait for someone to make a freaking like hierarchy chart? Or is this a filled in circle? Is this half filled in, or is there, you know what I mean? Like, nobody would. Oh, it's so frustrating. It's so. I mean, genetics nowadays is more like computer science than it is like, these Punnett squares. Like, it's just. Oh. But there it is. We have to teach Mendelian genetics and stuff like that, as if, like he was the first person to realize that like you could breed things that have separate genes and stuff. I don't know. I just think that it makes me so mad. Sorry, I get really frustrated this
No,
one.
no, I hear you. And like, yeah, it brings me back to sort of what I alluded to earlier of like, I feel more valued in the company that I work for than in,
Hmm.
and I do less wet lab work for the company than I do for my academic research, where like the majority of my wet lab bench work is like in the academic
Hmm.
setting, but I feel undervalued there, where it's like, yeah, they're not, the company's not gonna, pay me the salary that I ask for or that I command to like, again, adjust the pH of the buffers or like to fill tip box. Like, no, like we're paying you because we want like your expertise. We don't
Yeah.
care. We're not gonna have you waste time like, casting gels or like, weighing out some powder to make your buffer. Like, and that's where again there's totally some issues with industry that I have no problem talking about. But like, yeah, it's like it's almost like all backwards. And same thing when it comes to teaching, where it's like I've said it many times before, and I'm open to be wrong, but you know I sort of started to feel or like have my passion reignited for science in university when I was like took my first biochemistry course. And it's like, oh, like, I was asking myself the question at the time, like, why wasn't I exposed to this like earlier on? Why am I
Hmm.
only now learning about proteins and enzymes, etc., etc. Where like I feel like, some version, maybe some more like watered down version of biochemistry like can be taught at like even like lower high school or like elementary school. I think you can
Hmm.
start introducing like some of these topics. Uh, but it's just like, yeah, it's just like super dry. Um, and it's like, I think society is paying a price. I think understandably many people are driven away from science as a result of that.
It's very practical knowledge that it's not even just like becoming a scientist, which I 100% agree with you. Like, it's also just look at PCR. Like, for example, it's like that's a molecular biology technique. Why would you need to know about PCR? It's like, well, when you go on a jury, in the United States, like they'd spend 3 days to you telling you, one, what DNA is, second day, like what PCR is, and three, how do we actually know that this is what it is? Because that's what the murderer's DNA is based off of, right? The DNA analysis. So like it has real world implications too, especially with like, the pandemic and stuff, all this RNA. Like, how do we, and people are hearing about RNA probably for the first time in 20-30 years, and now there's an RNA based technology and they're getting it confused with my DNA and all that kind of stuff. And it's like, i I don't feel like I don't I mean, I can see why people in science has disconnected from society so much. I can see why people are like very cautious. They're like, "What's this new thing?" or "What's going on?" And because I mean, you have like a gap between like the layman, the high school and like undergrad, and then there's like the undergrad and like graduate students, like a huge jump where it's like we're doing stuff in research science now that's like legitimately science fiction, like legitimately. People wouldn't think that it was even possible. People wouldn't even think that there's just it's so crazy what's happening. And that's the stuff that I try to that's my goal, to be honest with you, for my channel, is that like I want to blur that line. I want people to think that it's science fiction. Like, we can't cure blindness, but we can, with nanoparticles that have viral little parts on them that can kind of like specifically go to the eye and cure genetic blindness in mice. Now
Okay,
we can
yeah.
do
So
that.
basically, gene
It's
editing
like,
with viral particles, if I'm understanding it
yeah,
correctly. Okay, yeah.
yeah, yeah, exactly. David Liu's lab, where he's like doing some like really crazy stuff for like, George Shurt's doing
Oh
in
yeah, I know
situ
George Church
sequencing now.
very well.
But in situ sequencing, so it's like there's actual like sequencing that's happening inside an organism, inside a microbial community, so we know, what kind of organisms is in there. Like, it's just it blows my mind. So that's what I get like super excited about, man. It's really cool.
Yeah, no, for sure. And so if I remember correctly, like you basically consider yourself, and your training is like in strictly or largely speaking microbiology. Is that fair? Or like
Yeah,
microbiology slash molecular biology?
It's funny. Like I've really my undergrad was in ecology, behavior, and evolution. So because I wanted to be like a park ranger, kind of like save the environment in that way.
Mm-hmm.
And then I did ecology labs, and I was like, I wanted to be out in the environment. I don't want to be in a lab, how, I'm going to be out there in nature. And then I did some ecology labs, and I was just like, this is awful. Like, I'm not having fun, like counting ants crossing a line in the sun. Like, I can't do this anymore. And then I found out that you could do molecular biology and still save the world, kind of thing. And I'm like, that's it. I'm doing that. And then I had like a stint in molecular biology, and then I pretty much relegated myself to kind of like microbes and plants. I don't want to kill mammals.
I'm not gonna lie. Based off of my education, my undergraduate education, and what I've been exposed to about plants, I hate plants. I hate learning about plants. I hate at least, but to be fair, all my knowledge like comes from whatever examples of like Arabidopsis and just like and especially when it came to understanding like the genetics of plants, it's just like I don't want to touch this. Like this is just like but maybe just like my brain is too simple to understand it. But I was like, I don't want to have anything to do with plants when it comes to studying them.
Yeah, I guess it's because like I find like biochemically the process of photosynthesis really interesting, and like kind of harnessing that idea. I think that like the future, I mean, the major unexplored area of biology is microbiology, for sure. Like that's 99.9% of organisms that have not been discovered within the microbial world. So I feel like that is the last unexplored place. So in the latest frontier, the last frontier. So I'm really excited about that. But like, so yeah, I guess my background's like really diverse. I see someone that's like, I go to seminar or whatever, and someone's done like 30 years of research just in molecular biology. And I'm like, whoa, like I'm like blown away, man. Like their understanding of mechanisms and what's going on. I'm like, geez, but I don't know. I like having kind of like a breadth of different stuff, that's just kind of like how I am as a person. So, but kind of, I guess it's kind of funny that my background is like that. But now, especially doing nanoparticles and nanotechnology stuff, I'm like, I got a chemical engineering PhD that I'm working on now, pretty much, dude. So it's like that was the whole thing that I didn't expect to go down that road. That's really crazy. So that's been kind of weird to stretch my mind in that way. So
so that's a pretty fun. Based off of like what you're doing now, just to maybe attempt to get a better understanding of the type of stuff that you're doing, are there sort of like when it comes to like your day in and day out kind of like wet lab work, is there like a top 3 or like top 5 like techniques that are like your go-to, or that that you perform regularly? I'm assuming it's not just like CRISPR day in and day out.
Yeah, I mean, like so I do, I'm doing a lot of like cell culture. I'm working microbes. I love microbes in general, just because they're easier to work with. You can grow so many. From a molecular biology point of view, like working with an algae, strain. Every algae is a plant. Think about like, a million algae per milliliter that I'm working with. Think of how much space I would have to have in order to plant a million plants. Like, that's just insane, right? So a lot of cell culture work, a lot of, we're not technically doing, for molecular biology wise, I'm, cloning with, I like, Gibson assembly, overlapping PCR stuff. I feel like that's really fun. Although everyone's like really big into Golden Gate and stuff now with the restriction enzyme building block stuff, which is cool. I just haven't had really good transformation efficiencies with them. I don't know why. Maybe I'm doing something wrong. I don't know.
I'm as of now not the greatest position to help you in troubleshooting with that, because again, a lot of my molecular biology, like since I'm a biochemist and like pretty much like the vast majority of my work is like protein based, like most of my molecular biology techniques, like I used in the beginning of like cloning. I got my plasmid construct, you know, the way I want it. I got it expressed, and now that's it. I can forget about it.
Yeah, it's you know, and now specifically I've been doing a lot of like nanoparticle fabrications, so coating carbon nanotubes with different polymers, EDC NHS reactions.
Okay, like amine coupling. So now you're talking my language.
Yeah.
You're doing, or yeah, some sort of like look. It seems like cross-linking or coupling chemistry.
Yeah, sometimes like we have to have a fluorophore that we need to bind to a covalently to a DNA piece of DNA.
Yeah.
So we'll do amine, it's funny. I didn't know what nanotechnology even was before I joined this lab, and it's actually kind of like, you're not using nano tools, you know what I mean? You're using just like filters and centrifugation, and it's all the same things, and sonication and kind of stuff, plate readers. I guess you're using a DLS, dynamic light scattering, to kind of like zeta sizers, trying to see what size they are. We're using a lot of confocal microscopy. That's been the big technique that I've kind of like learned. Yeah, dude, working with like this $1.2 million confocal microscope, that's just like, I mean, that thing is nuts. It's like the screen, it has this huge 36-inch gigantic LED that you're using, and it's in a computer screen, but it's like there's all these knobs and stuff, and all these sliders. It's seriously like, those cockpit of a 747. It feels like what does this knob do? What's this knob do? What's this knob do? But you got to make sure all those knobs are right, or you can't see anything. So that's blown my mind, man. That I'm used to microscopy being kind of like, looking through the thing and you're like getting a headache, and but that's not what actual microscopy is nowadays. It's so much fun. It is really cool.
I have very limited experience with confocal microscopy. I've only like very briefly dabbled with it. And any knowledge that I had back then is like, well, it never really took in my brain to begin with. But like, yeah, microscopy has always been sort of like on my list of things, of like something I'd want to be more exposed to, because it seems like a really powerful technique. And like, looking at fluorescent stuff under a microscope, always cool.
Yeah, it is fun, man. It's like one of those, it's funny. It's like one of those techniques that you don't really normally see at a biotechnology company kind of thing, because it's like not very, I don't know. I guess I can understand applications that they would have really expensive microscope, but yeah, it's a cool opportunity to like expand kind of my ideas. And I purposely didn't go into a molecular biology and genetics lab, because I kind of like had that as a background for like 7 years before going in, 5 at the biotech and then 2 in undergrad. So it's kind of like I wanted to try something different, and do something different.
I know exactly what you mean. That's one of the reasons why like I moved in the biochemistry direction, because like, I strictly did my bachelor's in cell and molecular biology and completed it. And when I was searching for labs to do graduate studies in, originally I was like looking at molecular biology labs, and it's like, it would have been, I don't know for sure, but I'm reasonably sure that it would have been like a much smoother transition, as in like all of the techniques that I have learned and been exposed to throughout my degree, like I'll be like applying them or learning more about them in these molecular biology labs. But my guiding principle and it might have been a mistake. Who knows? We'll see. But I was like, no, I want to like try something new, or at the very least,
Mm-hmm.
something where like, not that I'm learning something that's completely fresh, but like where it's less of a smooth transition, and hopefully show some maybe diversity in skills of like, oh, okay, like this, hopefully show that I'm not afraid of like going in a more different direction. Or it's like there's still obviously a life science aspect to it, but a lot of the techniques that I use, so it's been an interesting experience in terms of like conversing with other biologists, where it's like other biologists who have stayed in biology,
Mm-hmm.
and like now that I'm fully entrenched in biochemistry, and like the biochemistry or a lot of the biochemistry techniques. And so it's been interesting, kind of like there's observing like a clear gap, where it's like, hey, I'm using some of these techniques here that are like useful for you, but then the biologist like, I've never heard of this, and it's like, I don't want to like, if I don't know about it, especially when it comes to the biotech startup that I work with, and like
Mm-hmm.
my particular expertise of like SPR, like SPR totally has applications and use in the molecular biology world. And like our startup is like trying to like, we're basically like trying to convince like other molecular biologists like, hey, we can add value to your research. And it's like, no, I'll like stick with my current techniques. Like, no, your current techniques are like no good. But it's like, well, this is the devil I know.
Interesting. So could you give like a little overview of SPR, and then I kind
Absolutely.
of understand it as like at least the Wikipedia has it like a certain molecules has resonance, and then you can kind of like tune that in to like the molecule. But how would that have anything to do with molecular
Yeah,
biology?
that's an excellent question. And I don't blame any biologist or anyone for going to the Wikipedia page for surface
Yeah.
plasmon resonance and being like, this is just all gibberish.
Mm-hmm.
Even myself, who uses the technique. And I use it as sort of a biochemist slash biologist, as like I'm using it to answer sort of biology related questions. So,
Mm-hmm.
and here is where, one of the roles, or one of the reasons why the company brought me in is to maybe bridge that gap, when we're like talking to prospective biologists saying, hey, we got a biologist on our side. They can attest, like, this is a real technique. We're like every biologist, well, I think virtually every molecular biologist is familiar with something like ELISA, right? That's like
Yeah.
sort of like a bread and butter technique
Mm-hmm.
for very many molecular biologists. And SPR very much is in some cases a substitute, if not, depending on the case, better than ELISA. So
Oh, interesting.
to give you a brief overview, without getting into the physics, although I won't get into the physics just because even I myself am not qualified to go through all the physics of SPR. I basically just
Mhm.
describe it as spooky physics. But because like I'm not a physicist, and I've learned enough about the technology and the physics to just give you like a brief primer and to explain it in hopefully
Mhm.
a comprehensible fashion. But to get into like the nitty gritty details of like whatever electron resonance and whatever refractive index, all that stuff, like I'm not a
Mhm.
physicist. I still consider myself a biologist, and we are not physicists. We didn't go, we didn't pursue physics for a reason. So,
True.
um, generally speaking, the technique when it comes to the types of questions it can answer, it's bread and butter, or it's like major use case is determining protein-protein interactions. So that's so me as
Mm.
a biochemist, I work with proteins, and the lab that I work in, they're pretty much like strictly a protein-protein interactions lab.
Oh.
So what SPR does is essentially, you have a prism, and it's a prism. Actually, I can show it because I have one of these prisms right in front of me.
When it binds, when the protein binds, it's going to change its resonance, and
Exactly.
it's selective stuff.
You get a
Ah,
signal. Exactly. So
gotcha.
this is I don't know if it shows very well on camera, but you have a prism, okay?
Mhm.
And so light shines at this angle towards the prism, and you have a gold surface, gold shiny surface, and It's gold coated for a reason. Use gold specifically. So when light of a certain wavelength shines this prism, something basically, the spooky physics happens on the gold surface. And this spooky physics, what happens is it turns the gold surface into essentially a biosensing surface. As in, if there are molecules or biomolecules or proteins accumulating on the surface, yeah, as you sort of alluded to, you have a change in refractive index, and so light goes through, and then there's a detector on the other end, and you monitor that change in refractive index. And so in the output of the software, basically if stuff is accumulating on the surface, you will see a change in your signal. Analogous, the most basic analogy of if there are proteins interacting, you would see a change in absorbance if you're using a spectrophotometer, if a spectrophotometer could be adapted to doing that. So that's essentially its bread and butter. And so so we have there are many flavors of chips where pretty much it allows you to immobilize just about any protein you can think of. And one of the advantages, the key advantages of SPR is that it's real time. Okay, as I'm sure virtually every molecular biologist knows, with ELISA, you go through your dozens of steps, whatever, immobilizing, washing, blocking, blah, 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 whatever, titration. And then, you go to your plate reader, and then, you you just hope that everything turned out okay, and that whatever readout that you get, all the numbers, they're good. Where it's until you put it in that plate reader, you have no idea, at least as far as I can tell, you have no idea what's going on in that plate when you're doing the ELISA. And it's only if something is wrong with your data, then it becomes pretty difficult to tease out where in the ELISA something has gone wrong. Is it in the blocking step? Is it in the immobilization step? Is it whatever in the any number of washes? Whatever. Because it's what we call an endpoint assay. It's only after you've done every single step and then you develop the film, so to speak, that you actually see anything. As opposed to in SPR, once you put this chip in the device, you are shining light and it becomes a biosensing surface. You are monitoring it in real time. And so when you do immobilization, and the classic type of immobilization is through EDC NHS, through amine coupling. You activate the protein, and then with EDC NHS coupling, it'll attach to the surface. And so let's say, for example, you can put protein X on the chip, and then if you're asking the question, well, does protein Y interact with it? Well, then you flow over protein Y. And if there's an interaction and, the protein X that kind of serves as your bait, if it catches your prey protein, then you'll see an increase in signal.
Where does the signal come from? Is it that the bonds between the proteins themselves, or is the chip gonna change color as less things are attached to it, or?
Yeah, so you're asking great questions. So it's the change is coming from it's called surface plasmon resonance for because it's it really is, it's a change in the resonance, as in again, I'll have to go into the deep filing cabinet of a brain to extract the type of physics that it does. But yeah, when light is coming to the prism, it basically results in the electrons having a resonance signal. And so to answer your question of what is changing when stuff is accumulating on the chip, so there's no technical color change. It really is a change in what we call refractive index. Where
so is the metal itself
Yeah.
Is changing. Okay.
Yeah,
the refraction of the metal itself is changing as
it's
things
gold.
are bound to it.
It's specifically gold for a reason.
Okay, that's really cool. That's cool because it's in nanotechnology. We've gone to that's very similar ideas where a carbon nanotube will have binding potential for proteins, and if a protein binds to that carbon nanotube, you can basically have an excitation and an emission, and the emission will change, with or without the protein bound to it. if the protein is present and it does bind, then the emission changes, and you can see that with like an IR camera. You can see that with wherever the emission is. So it's cool because it's it's not tied to it. So it's like kind of like an off target or remote sensing is kind of like what it's called. So you could technically do it with like a drone or something. You could detect that emission change from like a drone on a plant or something like that. That's kind of the idea. So would this be like would you be able to do that? Is this like this has to be in a machine that's outside of other like light refracting?
So actually, well, here's where I will shill for my biotech startup. One of the advantages of our platform and our specific device is like, yeah, it doesn't need to be protected from light. Like it's out pretty much. It's not fully out in the open, but like, yeah, you can actually see the LEDs as they flash, like from the outside.
Oh wow.
And so like, yeah, cool. That is one of the advantages that we have, where like some other SPR platforms, they do have to be protected from light, although ours doesn't. But we're not the only one. But that is one of our advantages.
That's cool, because then you could use like a Raspberry Pi. You don't have to have a special microscope. There's a lot of now advantages where it's like
that's literally I'm not gonna give away trade secrets, but
yeah, I mean that's part of our a couple of our papers where it was the lab that I'm in's papers there. It's like using those costs. It's always good to use items that are cheap. Let's just say that.
Yeah, no, absolutely. And it's and again, another one of the advantages of not just my company's specific platform and device, but just of the technique in general is, like I said, it's real time and it's label free, as in you don't need a tag, as I sort of alluded to. Like if you want to,
big,
yeah, exactly. Where a lot of times where many other biologists or biochemists are trying to assess protein-protein interactions, like they're very often like modifying the proteins either chemically or adding a tag, etc., etc. And already like you've kind of like added in a confounding variable, and sometimes those tags are not negligible, or like it's not some small like his tag, and so it's like again, it's been so interesting sort of speaking to other molecular biologists, where it's like, we're coming in saying, hey, the ELISA you're doing, you're kind of, not only is an ELISA way longer, as in like from start to finish, a standard SPR experiment, let's say is on the order of, an hour to 2 hours. As opposed to like ELISA, I think like the bare minimum is usually like 2 to 3 hours, if not longer. And
interesting.
Yeah,
I would think that like there would be like a really cool application with like FRET too. So
yeah, fluorescence resonance energy transfer, yeah, or Forrester resonance, yeah, absolutely. That is totally.
So there's like a second, the primary reaction where it's like bringing that thing off of the metal, and then you have like, so then your resonance would change, and then you maybe could have like a secondary probe or secondary detection mechanism to see if that, like the thing that just got coupled or decoupled from the metal, would bind to another like secondary protein that would change itself. So then you could see if like it would be interesting for like drug discovery. I feel like using it something like that.
That's absolutely that's so totally. Not only the technology, but our device has specifically been used in drug discovery, where it's like, yeah, that is a standard experiment of like, hey, if you want to see if your drug actually binds to the target protein, well, just like put your protein on the chip and flow over that small molecule. And if there's binding that occurs, well, you'll see a change in signal. That'll show up in our software. And like,
very cool.
Yeah, they're totally cool. And again, it's been so interesting sort of speaking to other biologists where like they're so committed, or since I don't know, they're so attached to their own techniques, where it's like, hey, by the way, not only can I save you time, but like you can actually like see the experiment in real time, and that should maybe make it easier to troubleshoot if something goes wrong. But they're like, well, since it involves spooky physics, it's like, oh, I don't want to touch that. Since it's something like so foreign to them, even though pretty much like beat for beat, depending on the context, like SPR has advantages over ELISA, and that's published in the literature.
Mhm.
But they people will cling to it just because, like, yeah, they like what they like.
Yeah, yeah, exactly. I mean, it's like people still using fish, fluorescence in situ
Yeah.
hybridization, where it's like, I mean, it's okay. There's there's like a lot of different DNA like probes that are out there that are way different and way better probably CRISPR based stuff or so it's just interesting to see. Yeah, you're right. There's like an old guard in research science where it's like, especially working with nanotechnology stuff where you are you know it's kind of the same way. Yeah, I have to answer the same questions like, why would I use nanotechnology versus this, and it's like, well, you can, I guess you could do it inefficiently if you want to. You could plant those million plants like I was talking about before, but sometimes I don't know, it's I think it's just one of those things where like technologies like come together and there's like a like GFP for example in the late 90s that
Hmm.
kind of like being discovered, and then that's also the same time that like these huge wave of discoveries but like inventions and leaps forward and microscopy is happening. And it's like, wow, well look at us now, we have a whole new field of biology that's like springing up because of that. So I feel like it's rarely like one thing, one
100%.
breakthrough. It has to be multiple breakthroughs, and then you kind of meet in that new space, and you're like, "Are we best friends now?" Like so.
Yeah, exactly. And I would totally understand some biologists' hesitancy if this was like a brand new technology, as in like it's only like in the past like 5 or 10 years
Mhm.
that the technology has come to market. But actually, this technology has been around like for decades. This technique has been
Oh,
around
cool.
for decades, like pretty much like starting in roughly I think the 60s or 70s. It really only took off kind of like in the early 90s with like Biocore. I'm not sure. So, um, have you had had you never heard of SPR before
Mhm.
now or before recently?
Not until no, when your Twitter bio, when you were
Uh,
saying it, and I was like, "What is this?" So.
that's super interesting to me. So I guess maybe it's understandable if more of like your bachelor's is more in the ecology um discipline, where it's like, but I can even speak for myself. Uh, as someone who went through a bachelor's in cell and molecular biology. Like, I did not learn about this technique at all. I don't think very many other biologists are being exposed to techniques like this. Not to like whatever shill too hard for my own technology or my own company's technology. Uh, and
What?
obviously, you can't teach everything. I know that, but it's
I had the same thing with nanotechnology. I was like, man, this is like a joke, right? Like where like how this obviously is like science fiction. It's not real. Where because there was like the Avengers, like Iron Man's like my new suit's nanotechnology, and then you go into it and you're like, oh yeah, this has been a research field for like 50 years. Like and you're like, oh my gosh, like it's the Dunning Kruger effect. You don't know what you don't know, right? You have no idea, and then you figure it out, and you're like, wow, this is thing. And I mean, it just gets to like what we were talking about before, where we're not talking about the coolest things that are happening in any field at once. We're not teaching that, and that's where I feel like school should be, at least in the undergrad area. I would argue everywhere. I mean, I talk to my kids about like how cool photosynthesis is, and they're 5, so so
that's
and
great. That's fantastic.
so like I talk about chlorophyll, we talk about chloroplasts, we talk about algae, we talk about plants, like um you know I I I think it's just talking about, especially at the undergrad level, like talk about the coolest things that are happening, as opposed to this, we're sorry, dude, we're in 1700s biology still. We don't have the time to cover SPR, and you're like, well, then what's this for? Get them in at the cool stuff, and then they'll fill in their knowledge as needed. It's like I didn't really learn molecular biology. I took the classes. I took the labs. I worked at an undergrad, lab, and where I did Western blotting. I did some cloning, but I didn't really know molecular biology until they gave me my first project of like, hey, go knock out Q70 and Eroya Lipolytica, and I'm like,
Until you actually had to like dive in, sort of like a little bit dive into the deep end,
yeah,
and just like you got to learn to swim. Hopefully not
yeah,
that. Hopefully not
exactly.
that dire in the lab.
No, it's really fun, right? Because then you're like, now you're realizing, oh, like that's why every lab has bacteria glycerol stocks, because it's like it's a library of DNA, and what's really good at keeping DNA? Plasmid DNA, bacteria. Like, oh, what a coincidence, now you're understanding, putting these pieces together of these labs. And I've gone back to my old lab notebooks and being like, oh, I was cloning there. Now I understand why I'm doing that. I'm saving that amplicon and I'm putting it into a bacterial plasmid to save that for later. But at that time, I'm not
You just
going
had like
back to it.
you
I was
had
doing it one time.
you had the all the pieces of the puzzle like separated and like slowly but surely like you were assembling it uh to hopefully see the full picture. Not that the puzzle is ever done. Uh, but just sort of as
Exactly.
yeah, but just sort of again as another example of like scientists like hesitancy to like really, even though in the lab and when it comes to answering research questions, like we are very much explorers. Like we're trying to answer pretty much like uncharted waters. But when it comes to like exploring in other domains, to me one of the greatest examples. This might just be unique to me. But so do you still use paper lab notebooks?
No.
Okay. So,
Well, I mean, technically you have to, like, for legal reasons,
sure.
so like benchling, that's what my electronic lab notebook does. And I haven't used it since working as an undergrad in 2011. So I went to an electronic lab notebook that you print out and then you sign for patents and stuff at the biotech. You have to
Oh yeah, no,
print
I
it out. You
understand for
have
the
to sign.
biotech. Yeah, I'm talking about
Yeah.
specifically like in the academic
No.
setting. Yeah, so I started using lab notebooks like years ago, but virtually no one else in my lab uses them. They are still like writing on paper, like in a literal lab notebook. I'm like, what are you doing? This is like a waste of your time. Not only is it a waste of paper, and like I dislike it as someone who tries to care about the environment, but like there's no reason not to use an electronic lab notebook. But since like that's what they have habituated themselves to, it's like, no, I gotta cling to my lab notebook. Not only like are you also like wasting your time like cutting and pasting, like making your
Yeah.
your collage notebook, it's like
Exactly.
it's like a time waster. Where it's like and I was in my lab, I have sort of been redeemed because like prior to the pandemic, basically, I was the only one that I was able to access my lab notebook from home.
Oh, there you go.
There was a time where my university was totally like inaccessible, not locked down. And I said, hey, well, maybe you should have switched a long time ago to electronic lab
True.
notebooks. But even then, they are still hesitant. And it's like, you're a scientist. Like, I'm hoping you have at least some faith in in the cloud storage. This is not, the craziest technology out there.
I think the internet's going to be around for a little bit,
Yeah,
for sure.
I think so.
So, but I think when I was trying to make the switch to at the biotech, they did not want to give their IP to anyone. That was like their thing, where it's like we wanted to have full control, and like once our data is in another server, then there's like security issues. So then I just transitioned all my electronic lab notebook stuff to an Excel spreadsheet. Basically, I made an Excel spreadsheet that had 8.5 by 11 that I basically screenshotted my gels and all my data in, and then I just pasted it into that one sheet, and then that one sheet is my lab notebook that I printed out and then put it in. So it's just one thing to it's so funny that you say that, because before I did that, like once a week I would literally have like a scrapbooking session where I'm cutting out like gels and all like 10 different things, trying to like I want to put it all on one piece of paper, and now it's like, oh my god, it was so time consuming and awful. And then so it was so successful that I actually like the company like switched people. They were like, this is what Greg's been doing, so we want to we want to we want to encourage everyone to do that. So that was kind of cool. That they
Yeah,
kind of. Mhm.
like and I think I know that there are still others outside of my lab, at my university that like still use like paper lab notebooks just because like yeah, that's what we traditionally use. I mean
What do
maybe
you use?
right now I use since this is strictly my academic setting, I just use OneNote.
Mhm.
Like nothing fancy.
Oh, okay.
Uh I've entertained the idea of like migrating over to like Benchling, but I find like
Hmm.
OneNote pretty flexible and at least suits my like I don't have any like fancy needs. As long as I can like take notes and like copy and paste images, that's more or less like what I need, or like attach files. That's basically uh, all of the needs that I have for like a lab notebook. Nothing
Yeah.
crazy. Uh,
Well, I mean, once you go into like the cool thing about Benchling is that they have kind of like lots and these like registries that you can do, so you can kind of like create a list of entities that you have like so for example, I have nanoparticles or something, and I can kind of like have a nanoparticle registry that I can kind of like say, okay, this is it, and then all my other experiments that are attached to that one, I can kind of find and search, and you could do it with OneNote and Pages and stuff too. I've used that
Yeah, actually, yeah.
before.
Another advantage that I have found with electronic lab notebook, like you sort of alluded to, like it's searchable. As in, like
Oh yeah,
I have, a lot of notes in my lab notebook, and I don't remember like everything. In fact, I probably remember nothing from like 3 or 4 years ago. Or like maybe I just get a hint of like, oh yeah, what was my data, or like what were the results, and like it's easily searchable as opposed to like in a lab notebook. Like, oh my god. And also, oh, so many things to talk about when it comes to just like a simple notebook. Also,
it's
one of the
true.
reasons why I switched to electronic lab notebook is to hopefully whoever takes up the mantle of my research, the next grad
Hmm.
student that follows in the footsteps of my project, or like picks up the pieces when I burnt everything into the ground, uh, is that one of the headaches that I have encountered, and maybe this is just unique to me, is like I also like inherited the project. I'm continuing some previous grad students' work.
Hmm.
And it's like when I'm having like meetings with my boss, or when I had early meetings with my boss, or my PI, I guess to be specific, and they're like saying, "Hey, yeah, check 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 their lab notebook," and I go and check I'm like, this is useless to me. I can't read anything in here. Like
Yeah.
their handwriting is just uh like I pretty much like started from scratch to a certain degree of like I don't know why are we keeping records like this. Like why keep records at all if they're going to be records like this of like
True.
this. Maybe I just don't know how to read, I don't know, maybe I'm illiterate. But it's like this is just like an awful lab notebook, and like it's completely useless to me, and I'm just not going to consult it like at all. And so I'm hoping that whoever takes up my mantle is like, oh, they have something that's hopefully slightly better legible than whatever handwriting that I happen to have, and is searchable.
Yeah, man, it's the only way. I mean, I started loving my lab notebook specifically when I was trying to like reproduce things, like exactly what you're saying, like going back to a PCR that I was like, how do they not write the annealing temperature down? Like, what am I doing? And then, I have to do another gradient. I have to like figure that out, and then maybe it doesn't work, and now I have to troubleshoot it. And you're like, dang it. Like the lab notebook gives you an ability to kind of like say, oh, what's important in a PCR, and then kind of take a template and then say, okay, these are the criteria that I need to write down every time in order to reproduce it. And I really strongly believe that like people that don't care about lab notebooks or don't really write anything down, they're either not reproducing anything, or two, they really haven't been a scientist long enough in order to try to reproduce anything, which is very scary.
Yeah,
Like where
absolutely.
they're not like they don't care anything about reproducibility, and it's like that's one of the hallmarks of science for me, right?
Yeah,
Is
one
like
of the pillars, absolutely.
reproducibility. So to not care about reproducibility for me is just like, oh, you're not really. I mean, Adam Savage has like a great quote. It's like the only difference between having fun in science is writing it down. So
Yes,
you can,
exactly.
you got to be able to write it down so you can share that information for your manuscript or, for whatever. So, but hey, man, I've had a really good time talking to you. I gotta go do dad stuff. I
Oh
gotta
yeah, I totally understand.
do bedtime.
I can attest you have children that I audibly heard.
Yeah, this is my daughter. She, whenever I'm in the garage, she wants to come and like, especially the lights and stuff. They love playing with the lights out here. So it's been, yeah. But dude, it's good talking to you. Maybe we can,
There's definitely
yeah,
other
talk
things that
soon.
I want to touch upon. Specifically, I'm gonna let you go. I don't want to take up too much more of your time. But like, yeah, you alluded to like one of your goals of coming to this space is basically like science communication. And that
Yeah,
like you see like
yeah.
there's a gap in terms of like, yeah, there's a lot of like pretty much like poor at science journalism. And I totally want to
Hmm.
get your thoughts on not now, but maybe for a future
Yeah.
conversation of like, uh, I'm of the opinion that science journalism is like done very poorly, and like just like across the board, especially when it comes to like mainstream media news outlets. Like I just don't,
Hmm.
I would never consult them for any sort of scientific finding. And yeah, maybe in a future conversation, I want your thoughts
I
on those
totally
things.
agree. I mean, for anyone that I go to the news, I see that it has happened. I think it's cool. I don't take any conclusions from the science press. I see that it happened. I try to find the link to the paper. So many publications or
Never put
news
the original,
articles. Oh
absolutely.
my God, it's so frustrating. And you're like Google searching the institution and the last name, and you're like, okay, I found it finally. And then you can finally read the paper, and then yeah, I don't even read the science press stuff. It's really frustrating. Um,
And like, again, I'll let you go. But like, it's to your point of like the reference is never put, and like I'm even if this is coming from journalists with
mhm.
no scientific training, like to not put the reference of like an article that you're covering, like I don't like what are journalists being taught that's like they can't even
I
do
don't
that.
know. The worst up on it, by the way, I don't understand why, but the worst on it is the New York Times. The New York Times never sources the original study, and I think it's because they think that they're doing like a bedrock, like first person, like our news piece stands alone
Right?
kind
Yeah.
of thing as an
We should
article.
be the front
But
page, not the actual.
yeah, so it's like because we're doing original interviews, we're doing original reporting, so now we're the primary resource. When it's just, I don't feel like that's true. I think in general, just everyone should be reading primary literature. We've gotten so far away from that, and it's just so frustrating to me. Um, the amount of things that are just kind of like third hand, and that's why I like to go like on stream and stuff, go over like actual research papers, and that's not not the stuff that it does well. You know, when I'm
I know.
debating people and stuff, that's like that gets way more views than like the coolest groundbreaking science. But um I don't know how to make that transition, but hopefully people can kind of like see it, you
Yeah,
know.
but yeah, maybe hopefully in a future conversation, if you want to like hash out or cover more about science journalism, maybe what can do better,
Yeah.
what's currently go wrong. But yeah, I'll let you go and do your dad stuff because totally your children
Okay.
take priority. It's been a pleasure.
All right, Corey. Great meeting you, man. I'm really happy to get in touch with you and talk finally.
Likewise, and thank you for your patience.
Oh yeah, it's good, man. All right, talk to you soon.
Take
Okay.
care. Be well.
Bye.
All right. Well, that was great. That was, well, despite the few hiccups in the beginning, that turned it out amazingly. That turned out amazingly. And welcome, Why So Dankness. Welcome, Chew Toy, Billy Bob Thornton. Missing a bunch of people. And also, shout out to Bill Bob Thornton. Oh, I know. I know your channel name. I'm one of your mods. How can I not know one of your channel names? But yeah, that was a great conversation. And I think I solved my kind of like streaming issues in the beginning. I think as a result of me updating OBS, it like changed my bitrate it increased my bitrate. I think I had my bitrate set much lower than what the update enforced on me. Oh, and Ultimate Strizzle, welcome. Thank you so much for passing by. I'm doing well. I definitely know you from many other channels. How are you doing? And yeah, there were totally many other topics that I put down that I wanted to cover. Hopefully in a future conversation, we can have that because there's totally a lot of things about science journalism